Welcome to Biomarkers 102, a follow-up webinar of Biomarkers 101. We are pleased you can join us today for this very special event. I am Erin Sullivan, Executive Director of Sisters Hope Foundation and Cure ALSP. We are the Global ALSP Foundation. Please note, we are providing the Biomarkers 102 webinar for educational purposes only. For any treatment decisions, please speak to your treating physician. Today's webinar was previously recorded and will be available on the Sisters Hope Foundation website. If there are any questions after the webinar, please direct them to info@sistershopefoundation.org. Today, we are pleased that Dr. Fanny Mochel, the Director of Reference Center for Neurometabolic Diseases in Paris, is joining us to answer ALSP community members' questions about biomarkers. Thank you, Dr. Mochel, for taking the time to share your experience and expertise. Let's get started.
Thank you for having me.
You're welcome. So the first question from our community is, can you explain again, why do we need biomarkers?
Well, biomarkers can be used for different purposes, so it's important to, first of all, clarify what we are using them for. Sometimes we are using biomarkers for diagnostic purposes. For example, in a well-known disease called Alzheimer's disease, we are using biomarkers from the lumbar puncture, from the cerebral spinal fluid, to measure some proteins to know if there is a profile that could be evocative of this kind of neurodegenerative diseases. And therefore, in this case, and then we have other examples in the dementia field, where biomarkers are actually helpful for the diagnosis. In the case of ALSP, we don't really need biomarkers for diagnosis because we have the genetic testing available that is going to give certainty about actually whether or not a patient is affected with this disease. Although I must say that sometimes when the disease is not known in the family and that we're exploring a patient for leukodystrophy and we find some what we call a variant, so a change in the code for the gene of CSF1R, sometimes this variant has not been described. And therefore, I mean, in other patients affected by the disease. And so then it becomes what we call a variant of unknown significance. And in this case, actually, it would help to have other biomarkers that could tell us this is really pathogenic. This is not just any variant, because we are all carrying hundreds, actually, of variants, meaning some changes in the code that are rare, but not causing a disease. So we call them benign. But of course, what we are looking for here is a pathogenic variant, a variant that is going to cause a disease. And sometimes, again, when it has not been described, it would be useful for ALSP, but again, it's a small number of patients that are in this situation, to have another kind of diagnostic biomarkers. And for now, we don't have that really available. When we are usually wondering about this, we are looking closely at some MRI of the brain, so imaging of the brain, because we know that there are some aspects of the disease that are evocative of ALSP, of course, some of the clinical presentations, but these are going to be combined arguments, but we would like sometimes to have an additional diagnostic biomarker for the unknown variant found in the CSF1R gene. That's one kind of biomarker. The other one that we are, I guess, here more interested into are what we call the prognostic biomarkers, the one that are going to inform about disease progression, about disease evolutivity overall, and of course, response to treatment. Because when we are going to conduct a clinical trial, usually, we don't run a clinical trial for years and years, 5, 10 years. That's usually not the format of most clinical trials. Therefore, when we need to have an answer early on, usually after 1 year, sometimes 2 years, unfortunately, the clinical evolution of patients sometimes is not sufficient. And therefore, we need to have other aspects than clinical examination or some cognitive testing to actually know if we are hitting in the right direction with treatments. And for that, we actually really rely on biomarkers. And so for ALSP, these are especially biomarkers from brain imaging. So not the classical white matter abnormalities that usually are not very, very progressive over time, but other kind of biomarkers. Maybe it's a part of the atrophy, the brain loss that goes with the disease that we try to measure. Sometimes it's new tools to actually measure all the white matter content that we cannot just see with our eyes with the lesions, but something that we can measure in percentage very precisely. So these are the kind of biomarkers that we are looking for, because they will be very useful to know if they can be changed from their natural history when we actually apply a treatment, so that we can have an early indication if the treatment is going the right direction. And also because if we have a sensitive biomarker, we do not need to enroll as many patients usually as when we have only clinical readouts, because every patient is different. So for one patient, it's the cognition who may be interesting. For another patient, it's the behavior. For another patient, it's some motor score. But to grasp that in only one item is very hard. So that's why when we have something more homogeneous, like brain imaging or other markers like the one we can take from blood samples, and particularly we have something called the neurofilament that we take from the blood, but also from the cerebrospinal fluid. In leukodystrophies, like ALSP, but others, we know that this is also usually associated with disease progression. And if we see a sharp increase in the NFL levels, it's usually of bad prognosis. If we see something that is slowing down or actually even being stable, it's very encouraging. So it's really mostly for clinical trials that we need really those biomarkers to counteract the main clinical heterogeneity that we have. And so that we can also have trials that are going to last for years before we have an answer of whether or not the drug or the intervention is useful.
Great, thank you for that very thorough answer. The next question is, and you kind of hit on it, but I think that it bears repeating again, is genetic testing a biomarker?
So actually the definition of biomarker can be very broad, because in the end, it's a marker which is biological. So you could possibly call that a biomarker, but we tend to use the word biomarkers to really see what can go along with a disease that we know the patient has. So for example, in the case of ALSP, the genetic testing is really what is going to provide certainty about what is the diagnosis. And what the biomarkers that we are looking for are rather all the other parameters, usually imaging, but possibly biological, like NFL, that can inform about the disease progression and the response to treatment. Because the gene itself is not going to tell us what kind of disease is going to develop, when, what evolution, or what response to treatment. So that's why we don't really call it a biomarker.
It's just used for diagnostic purposes.
Exactly.
Great. Okay. This one, I enjoy this question. I think it's going to be really interesting. What biomarkers do you commonly use in your practice?
So in my practice, right?
Yeah.
Okay. So I guess we because I'm a reference center. I have a reference center, sorry, in France for neurometabolic disorders, but also actually for leukodystrophies. We have actually a lot of history in France with centers for rare disorders. So we have kind of the luxury to be very specific for the kind of diseases that patient may have. But for leukodystrophies, like ALSP, we actually tend to have some kind of common thinking. So not for all kind of leukodystrophies. There are very different aspects. For example, some of the patients that we see have rather a problem of the myelin that is not going to take place and develop in the brain along childhood and adolescence as it should. We call that hypomyelinating leukodystrophy. This is a very different picture than what we call a demyelinating leukodystrophy that starts in adults, where the myelin that is there is going progressively to be attacked and to be damaged, and therefore to have consequences. But if we look among the demyelinating leukodystrophies, Again, they are not all the same, but we tend to have similar ways to actually treat them and also to follow them. And in particularly, we do have similarities with even more common leukodystrophy that is called X-linked adrenoleukodystrophy that affects mostly the men, unlike ALSP. And therefore, my response is really combined for what we do for patients, for example, with adrenoleukodystrophy and patients with ALSP. So what we do, because we know that for these two diseases, actually, they have in common that the immune system of the brain is impacted by the mutation. In the case of ALSP, it's mostly the immune system that we call the microglia in the brain. In adrenoleukodystrophy, it's not only the immune system, but it's an important component of the demyelination. And so for these two diseases, for example, we have experience with the transplant as a treatment, and where we replace the immune system of the body to replace the immune system of the brain. And therefore, for that, we are going to monitor aspects of neuroinflammation. So we use in our center imaging biomarkers that are based on a method called diffusion tensor imaging, which is looking at the propagation of water along fibers of the brain. And depending on those propagation parameters, we can infer metrics such as the number of fibers of the brain, or how atrophied the fibers are, in particular, or if there is some kind of inflammation. So there are different parameters that we can extract from this diffusion method. And we think that for leukodystrophy like ALSP, but also like adrenoleukodystrophy, that these are actually very sensitive to change. And that hopefully with this method, we can detect changes before the evolution of the classical lesions. That usually when we see that, it's a bit late already in the process of inflammation and demyelination. So this is one kind of biomarker that we use a lot, again, to try to not just look at the lesion, but the whole white matter of the brain, and not to look at it only when there is a lesion or several lesions, but at the early stage. And this is, of course, particularly true for when we detect the disease at the early stage, especially, unfortunately, not with the first patient in the family, but when we know that we can monitor others who also have the mutation. So that's one very important tool for us that we are using. And when we are performing treatments with transplant or other, like in the VIGIL trial, for example, for LSP, we are going to use these diffusion tensor parameters to actually see if we have a global response on the white matter. Another one I already mentioned that we are using quite a lot in this leukodystrophy, in this demyelinating leukodystrophy with loss of myelin, is also NFL, this neurofilament light chain that has been known for many years already in the field of neurology. Because it's something that is abnormal with any kind, I would say, of neurological disorders. But it seems that in the leukodystrophy, it's really sensitive to change regarding actually that aspect of inflammation that we want to target for treatment. So usually really that marker is going up when the disease is really evolving or actually accelerating or at the onset of symptoms. And it stabilizes when we have treatments that can halt the disease. So at the moment for us, it's really these two biological and radiological biomarkers we are using, but not only in ALSP, in other disorders where we have more actually history in treating and detecting.
Wonderful. We've got a couple of questions on NFL. So the next question is, will my primary care physician be able to look at my NFL?
So unfortunately, no, because at the moment, really, first of all, I mean, in terms of the value is abnormal, possibly, yes, any physician, if there is a normal range, if you have a measure, if the measure is above the normal range, would say this is abnormal. But there is not sufficient knowledge for now the general community of medicine and even of neurology to actually know what this elevated level is saying. Is it mildly elevated and relatively stable and therefore not informing about so much disease progression? Is the level of elevation actually already an indicative of something that is soon going to progress? It's really when we look at these values in a cohort of patients with the same disease, or if we have several time points, that we can actually also correlate with the images, because it's almost never one parameter out of context. It is important to know if there is any other factor involved. I mean, for example, for now, we don't know really if another kind of inflammation could interfere with the NFL level. Let's say the sample was taken during like the start of a flu or any kind of viral infection. Maybe this is also impacting on the level. So really one value in isolation is not going to be interpreted. And so it's really the expert centers who have several time points, who have not only the NFL, but also the imaging evolution of the patient with the clinical aspect, that can say, yes, this is unfortunately telling us that the disease is progressing, and maybe if we have a treatment at hand, it's now that we should use it. Or for now, this is not something that can be interpreted, and therefore we keep this in mind, but for now we don't take action. So, but maybe this will change, and maybe when we have, of course, more experience with these markers, as we are starting now, for example, in leukodystrophy, in the children in particular, there is now a cutoff above which really the pediatrician know, okay, this is becoming really an alert. We are not there yet with ALSP. We need more time, we need more expertise, but maybe in the future. But for sure, at the moment, it's not possible outside of the end of an expert.
So what I'm hearing you say is it's really important, especially in ALSP, to use all possible known biomarkers working together to understand the disease and the disease progression and possible treatment options.
Exactly.
Okay. The next
And
question.
if I may, sorry, just add on what you were just saying. Usually when we try to provide with the best treatment option, in most centers, I would say it's more or less never the decision of only one physician. It's usually a discussion between different experts even to know what's the best course of action. So it's different markers, but for disorders like that, for which we are still exploring the best therapies for, again, a given patient at a given time, it's really a matter of different experts trying to agree on something.
Thank you for that clarification. The next question is also about NFL. Will a biomarker like NFL help to determine why one person with ALSP progresses faster than another person?
So, yeah, it's an important clarification. I guess that the NFL course of evolution, I mean, I guess it's not one time point, but several time points that we can take into account. This is going usually to not so much, I'm not sure we can use the word predictive because we are not there yet, but it's really indicative that something is going on and that we should be more careful at the MRI because maybe this is, or at some cognitive changes or something that tell us that we need to offer treatment, for example, for the people who are carrying a mutation and have not yet developed symptoms. So for that, again, it's an indication of disease activity. But the elevation of NFL is not saying why this person actually is more prone to develop the disease. Again, it's more like the tip of the iceberg. Like, yes, this is changing and careful. I mean, the disease may start now in terms of demyelination, inflammation, but it doesn't say why. And it doesn't say why we have different disease onset, different ages for disease onset. It doesn't say why some patients will never develop it versus others. These are other actually parameters that we are looking at, our team in Paris at the Paris Brain Institute, but other teams obviously as well in the United States or other groups in Europe, in the UK. And some of us are looking at expression, gene expression profiles to see if there are other markers that actually may indicate that there is a trigger for the disease onset. Some of us are looking at the immune cells themselves, because the ones carrying the mutation in the brain, we can have them just in the blood, and these are called the macrophages. And so our group in particular in Paris is looking at that. Can we have some indication, because those cells are very accessible through a blood sample? So there are a lot of other things that we are looking at to try to understand what can be the trigger. And sometimes we also wonder if it's not an environmental trigger. It's true for ALSP, but it's true for also other leukodystrophy like adrenoleukodystrophy, that sometimes a head trauma, not any mild head trauma, but like a car accident or something, it's peculiar that we hear that the symptom actually happened soon after this kind of thing, just because this kind of impact we know is going to have some activation actually of the immune system of the brain. So if it's already the immune system is already fragile because of the mutation, and you had something that is going to activate it, well, if you activate something that has a genetic defect, maybe this can be a trigger. But it doesn't mean that all people carrying mutation in the gene CSF1R with a head trauma will have symptoms. That's not what we are saying either. But it's maybe not only genetic modifiers, but it can be also environmental or other things that we are still exploring.
It sounds like your team is exploring a lot of different avenues, and we're definitely thankful for that in our community. All
Thank
right,
you.
couple more questions. The next one is, does looking at biomarkers help to find treatments for ALSP?
That's also an important question. This is what happens sometimes, indeed, that we are trying to find a marker that will evolve with the disease, which may be indicative of symptoms onset or disease progression. But by doing that, we actually identify something that can be a target for therapy. So it is what is also very important in this research field, to try to grasp what can be the other factors that we can look at beside the brain, especially when it comes to biology. Again, the NFL so far is not really giving us so many ideas, because we know it's a marker that is going to change with degeneration overall, and in neuroinflammation, it's very sensitive, so it's going to go high. But it doesn't say much about what exactly is the mechanism that is making ALSP progressing. But other markers, for sure, that we are looking at, other inflammatory markers or genetic markers, these can definitely be a way to offer novel avenues for treatments. And that's why it becomes so important. And this is true for ALSP, but for, of course, also many other disorders.
All right, and the last question, does looking at biomarkers help me or the drug industry?
Well, it can be both, and we hope it's both. And in the sense that participating to studies where we are looking at biomarkers can help refine which biomarkers are the most predictive of, again, symptoms progression and response to treatment. And in this particularly, in this case, when there is a trial, actually the biomarker that you have been helping to develop or to refine is going to be the one which is going to have a determinant effect for starting a therapy in yourself or monitoring a therapy in yourself. So this is a kind of direct benefit. And we have patients and other disorders on which we worked on where we actually were able to identify this kind of biomarkers that then were helping for the clinical trials that we run in order to assess therapy and give us actually ideas for therapy. So it's, I would say, really a direct benefit. But then, of course, there is also what we said before, in the sense that if some academic group or some industry come with a new idea for treatment or wants to explore further the benefit of existing treatment, and they don't have the right parameters to look at, Well, it can easily be, it can easily lead to the situation where the study is considered negative just because we didn't look at the right marker. And that's a really, really important thing to understand when it comes to neurological disorders in general, but in particular for rare diseases. And like colleagues, I've been involved in discussion with the FDA, with the EMA, and in actually situations that are very sad, where you have evidence that a treatment is helping, but we didn't anticipate enough what was going to be the primary outcome measure, in particular the one on which we consider this is a positive or a negative trial. And if you don't have enough data on that before the trial, and that you miss your target, especially what we call the primary outcome, the one on which we decide if it's positive or not, well, then all the development that has been done can be just thrown to trash, because the agency would say, no, sorry, it's a negative study. And in the meantime, yourself, you have a lot of other evidence that, no, this is actually encouraging and should be pursued. But it's very difficult to go back to those regulatory agencies that are the end the one deciding if treatment become accessible to patients. So really, it's a work for all the community, from the patients to the family, to the physicians, to the researchers, to the drug developers, to try to grasp as much data as possible to refine what can be the most sensitive markers, the one that are going to really change if possible in short time, so that we have answers in, again, not too many years, so that then we can discuss with the agencies, okay, we really think that if this is going in this direction, we will have an answer about this drug, and it will be one step to further develop and at some point to get access to the drug. So it is actually critical, and we have experience where unfortunately that could not have been done, and that we cannot further explore a very promising therapy.
It sounds like, as we all know, but you said so well, you know, understanding disease onset and disease progression and what therapies may or may not work. There's a lot of different components that have to come together, and a lot of different key players in that, from the patients to the researchers to the pharmaceutical company, to just try to understand all the mechanisms of the disease, which are quite complex.
Yeah, exactly. And especially because when it's rare, obviously, there are less patients, and so less data. So each little piece of the puzzle, but analyzed in a standardized way, systematic way, with everybody contributing, this is when actually it becomes possible to make science out of it, and not just saying, oh, it's rare, and that's why a lot of things are changing in the field of rare disease, which is very, very, very encouraging, because now it has become really, especially in the field of neuroscience, a field where a lot of efforts from the academia, from industry are being done. And it becomes even more than what we see for more common disorders, where, people are less actually driven to know where to start, where to go. Here, at least we know where to start. And communities usually are also better connected, I would say. And in that sense, they may advance way more. And so I really believe that we are at the beginning of seeing those changes in the field of rare disease, but it's going to be exponential, which is really great to see.
Yeah, it's really wonderful. It's really wonderful. Well, thank you again, Dr. Mochel, for your time. We truly appreciate your thoughtful and thorough answers, and using your expertise to help the ALSP community.
Thank you for
And
the community to also share with us, and for the really great questions.
And thank you all for submitting your questions and joining us today. We hope you found Biomarkers 102 helpful.